Hello.
Hello, everyone.
Good morning, everyone.
Welcome to our audience from around the world, and we're at the Research Panel Q&A. I'm Pam Bauer, Vice Chair of the MSA Coalition Board, and I'm joined today by my board colleague, Dr. Larry Kellerman, who will help facilitate audience questions. Please type your questions in the chat box on your screen. And answering your questions today, we have Dr. Vic Khurana, Dr. Daniel Clausen, Professor Gregor Venning, Dr. Wolfgang Singer, and Dr. Dana Wusman. Thanks so much to our distinguished panel of speakers for volunteering your time to be with us today on a Saturday. So we really appreciate this. All of the talks today were really interesting. I hope everyone had a chance to view the talks before you joined the Q&A. I'm looking forward to some really stimulating discussions as we answer your questions as you guys answered the questions from the MSA community. Larry, could you go ahead and pose the first question from our audience?
Yes, let's start with one. It was posed early in the panel. It says the other day a speaker said that brain research has shown that 40% of those diagnosed with MSA were misdiagnosed. Do you think this might be because not all MSA patients have run through a full battery of tests for their diagnosis? I don't know who that would be going to. I'm guessing the first one would be Vic and or Daniel.
Or Gregor.
Or Gregor.
Yeah, I can start. I think, I'm sure my colleagues will have some opinions on this, but I think one of the challenges that you run into with MSA is one, that you perhaps don't diagnose it earlier enough, or two, that it gets diagnosed like with Parkinson's and then later on, the symptoms change and that person ends up having MSA. So sometimes it can be very difficult to get to a clear diagnosis. I think that in 2022 we're a little better at figuring out who might have MSA and who might have, say, Parkinson's disease or Lewy body disease based on some new biomarker research that's going on. For instance, you can do these things where you look at alpha-synuclein amplification assays. You're able to see a different pattern of fluorescence with an MSA profile than you might with, say, Parkinson's disease. There's also some research looking at other imaging or other biofluid biomarkers. So I think while they're not ready for prime time for clinical work, they're closer to being ready for prime time. So I think a lot of research studies will end up having maybe different assessments or different assays that you would get as being part of a research study than you would for a clinical study. But I do think that generally speaking, the road to a diagnosis in MSA is often a long one, because sometimes the symptoms are really subtle or they're not really clear-cut clinically. And so I think as a community, what we're trying to do is to get to an earlier diagnosis, because those patients may benefit from disease-modifying therapies when you get them earlier. But it's a very excellent question, and I think points to the nature of the difficult diagnosis, but also the need for interdisciplinary care for MSA patients.
Dan, I also think that it's important to emphasize that study that found that 40% misdiagnosis was a brain bank study, and they accepted brains from community neurologists from all over, not from experts necessarily. Our data here at Mayo, and I know from several other centers with special interest in MSA, the data look very different. We have a 96 to 98% correct diagnosis rate. And so I do think it has in part to do with the rarity of the disease. And then again, I think with expertise, with seeing patients on a more regular basis and having additional tests to support the diagnosis, like, for example, autonomic testing, you can really improve the certainty of diagnosis considerably.
I would like to add to that. When I started, there were very few neurologists who knew about MSA. Neil Quinn was very important in that regard when he published a major review 30 years ago, but many neurologists didn't know what MSA was. I think now, if I look at the textbooks of neurology in Germany, actually, uh, MSA is a firm part of the textbooks of neurology. And I would say that, we have 8,000 neurologists in Germany. I would think that all of them now know what MSA is, but they only know the full house. When it comes to early stages or prodromal MSA that we defined in the criteria, the first symptoms and signs of MSA, there is an incredible confusion. And as Daniel pointed out, the early differential diagnosis is really what matters these days for protective therapies. And if we are not able to improve on that in the early stages, I'm talking about the first 1 or 2 years after symptom onset, that's what we would like to achieve 100%, accurate diagnosis. And we are very far away from that. We need a lot of more research in this area, which needs to consider wet biomarkers that have been studied by Wolfgang Singer a lot, but also MRI that has been studied by Daniel. And maybe, I could even think of, modeling prodromal MSA like Vic does in the humanized stem cells. So I think if we don't make progress in the early prodromal MSA patients, we will never be able to generate a cure for this disease.
I agree with everything my colleagues have said. I think that early diagnosis is critical, and I think that in the last couple of years, as Daniel alluded to, we've seen some really impressive developments. In my talk today, I ran through a battery of tests that are very closely aligned with the new consensus criteria by Professor Benning and others. And I feel that we have a range of tools in 2022 that we didn't even have 2 years ago. And I'll underscore what Daniel said. Again, we actually routinely do skin biopsies now, looking for alpha-synuclein, differentiating some of the harder cases from another closely related disease, progressive supranuclear palsy. That test really wasn't clinically available a couple of years ago. It's now done routinely in our clinic. And as Daniel said, increasingly we are amplifying alpha-synuclein. We even had in the last year a very exciting press release from a company, AC Immune, saying that they had a radiotracer for alpha-synuclein, for MSA alpha-synuclein in the brain that the Alzheimer's field has had for some time. So this is an exciting time. We need to do a lot more, but I think we do have some tools that are really bearing the fruits of research in the last few years. We're seeing progress made.
I would like to, I have to confess that due to a family health issue, I need to drop out here in 10 minutes. I have to attend to that. I just want to mention one additional aspect. I think that we need to recognize, despite our ambition to detect early stage MSA, we need to recognize that maybe MSA is among those degenerative brain disorders where we still need post-mortem to verify the clinical diagnosis. If you look at some of these studies, one of them came from Jacksonville, another one came from the Brain Bank recently. they all show that we are really terrible with the diagnostic accuracy, even in the final stages when we believe we are dealing with full house MSA because all the symptoms are there. We may be misled in up to 40% that, about the diagnosis. So I think what we really need to do is what Vic is doing in Boston, which is, focusing or zooming in on a nice cohort of patients with a diagnosis of MSA, obtaining, all these skin biopsies and biomarkers, MRI, and also PET scans, and trying to correlate that with post-mortem in as many patients as possible. I think that's the only way to really validate these biomarkers eventually. Because if you just use the clinical gold standard, I don't think that this will be sufficient, in my view.
Great. So moving on, there's a question here from Rich, who asks, are there any treatments that have proved to stop or slow the disease? And a follow-up, is stem cell or deep brain stimulation at all effective?
Well, regarding deep brain stimulation, there is clear evidence that this is contraindicated in MSA. It just doesn't work. And regarding stem cells, I think there is nobody more destined to answer that than Wolfgang.
Well, we are in the midst of a phase 2 trial to actually prove efficacy at this point. The data we have are limited to a relatively small trial from Korea, which was positive, and Mayo stem cell data, which were an unblinded, more of a safety study. So we have to be cautious to conclude that this treatment works. We need to prove that it works. And we need to do that in a controlled fashion. Now we are doing that in a study to exactly do that. So hopefully we'll have an answer to that question fairly soon.
Just one thing for folks to know is that I get this question sometimes, that there are stem cell trials going on for Parkinson's disease, and how is that different from what's happening with Dr. Singer's group? And just to make it clear, there are very different types of stem cell therapies. And so in Parkinson's disease, we're seeing the replacement of dopamine neurons, which is actually putting the kinds of neurons that are lost in Parkinson's back into the brain. And that is unlikely to work, unless we learn something very new about MSA. That's unlikely to work for MSA because MSA involves many different types of neurons that are getting affected and glial cells in the brain. And Dr. Singer's approach is different. It's introducing mesenchymal stem cells that create supportive factors for the brain. And that's potentially a very exciting avenue where multiple types of cells are being affected in the brain. So I just wanted to make that point. It's a question I get a fair amount.
I think one question, did I get that right, Pam? There was that question, is there any treatment that works? What did you ask that also?
Yeah, is there any treatment out there today that has been proven to work?
Well, one thing is clear, in my view at least. I mean, I might be narrow-minded because I just work in Austria as a small country, but I think as far as I can see in the MSA field, today there is no intervention or drug that can be prescribed that stops MSA. So we just simply don't have anything in our hands. At the same time, we recently published a review that also involved Pam about interventional therapies in MSA in the Horizon And if I count them up, I think there are about 30 plus interventions that are currently studied preclinically or, in early clinical development. But I think out of those 30, there's only one that I think is very exciting. And this is a small compound that's known as ANLI-138B that was developed by a group in Munich and is now commercialized, or it's in the process of becoming commercialized. There is a small company in Germany called MODAC, and they are kind of working with the Munich team to develop that compound, which is actually blocking oligomerization and fibrillation of synuclein and any kind of proteinopathy, including Alzheimer's, prions, PSP, Parkinson's disease. It seems to work for everything, at least preclinically. And so currently there is a Parkinson's disease trial, and shortly there will be an MSA trial. And they teamed up with Teva to co-develop this compound. So I think if I had MSA, I would try to get hold of that compound. To me, the evidence seems to be very strong that it might work in early stage MSA. For the rest, we just simply don't know, and we have to be patient and wait for more results.
So I'm going to temper Gregor's enthusiasm by making a point that I talk to my patients, and that is clinical trials are not clinical therapies. And I think one of the biggest frustrations patients have is they say, if I don't get in this clinical trial, then I'm not getting access to a treatment that may help me or cure my disease. In many cases, in most cases, clinical trials fail. They don't end up working. Doesn't mean we don't try. It doesn't mean we don't do more trials. It doesn't mean we don't get involved in research. It's just a thing that I want to say that a lot of times patients equate being in a trial with having a cure. And I think from a researcher who takes care of a lot of patients and who helps them go through this difficult journey, I want to make the point that it is research, and it's and very infrequently we have a result that's slam dunk. But just part of the point that I want to make. Sorry.
I wouldn't agree more than with what you said now, Daniel. If you look at the field of motor neuron disease or Lou Gehrig's disease, there have been 800 trials, and all of them failed. Although there was promise preclinically, in a lot of instances, clinically they all failed. And if you look at the stroke field, once you have the damage, nothing works. There have been thousands of compounds that were tested for stroke protection, and nothing works. And so, if we are pessimistic, we might think that since MSA is so kind of rapid and progressive and kind of aggressive compared to Parkinson's disease or Alzheimer's, it may also be resistant to these types of therapies in trials. But I think, we should give a sign of hope. That's why I mentioned this compound, because, you need to have a sign of hope that maybe one day things can be changed. And if I look at the entire field of what's going on right now, that's the only thing I wanted to say. I think the preclinical evidence base, of all these compounds is most robust for this small compound from Munich, in my view.
And I just want to make a plug for good research and understanding the disease. We have seen a phenomenal research effort in ALS in the last decade, and it has been funded by a variety of sources, but many of you will know that the Ice Bucket Challenge was a great catalyst for research in ALS, and the ALS community, to their credit, really created a very coherent, collaborative international community devoted to understanding the disease and the subtypes of the disease. And I think we are seeing, if you read between the lines of some of the latest studies in ALS, we're starting to see that needle shift finally for ALS, specifically for patients who have subtypes of the disease where there's a genetic understanding. And many people are trying to make that even better. So I think we're starting to see, I don't want to, again, like Daniel says, present false hope here, but we are seeing the needle shift with a variety of neurodegenerative diseases and neuromuscular diseases. It's an incredible change, and sometimes change can happen quickly, and we don't know where that major advance is going to come from, but we do need to understand the disease that we're treating and the subtypes of patients that have this disease. So I don't want to put a timeline on it, but I certainly want to convey that there's hope in research.
Larry, do you want to take the next question?
Larry may have a connection
We've
issue.
lost Larry. Okay. So I'll read the next one. Is there any doctor looking into the possible connection to exposure to environmental toxins and MSA? If so, who?
So I'll take that one.
Thanks.
This is a great question. We get it a lot. I'm sure all the clinicians here will agree that, why me? Why did I get this disease? It doesn't run in my family. Is there something that I was exposed to? For a very rare disease, it's hard to do the kinds of studies, we call them epidemiologic studies, to get very robust data for a disease like MSA. Fortunately, there is a roadmap for other diseases that has now been carved out quite nicely, where quantitative exposure information is related to disease risk. One of the pioneers in this field has been Dr. Beata Ritz at UCLA, who has really kind of transformed the way we think of environmental toxicants and Parkinson's disease. So a couple of years ago, the MSA Coalition funded Dr. Ritz to lead what we call Core E for environment for the coalition, and she's made great progress in taking the same paradigm that she's used for Parkinson's disease, and is generating a catchment of information through databases, environmental information in the California area, so that we can start to build the kind of records that she has used very successfully for Parkinson's to really answer that question for patients. So very important research. And you can see the fruits of her labors really kind of there are changes in public health policy now related to some of her work. Recently, we had a paper with Beata where we could take all of the toxicants that she's identified for Parkinson's and introduce them into human dopamine neurons to understand for Parkinson's which of these toxins are really affecting the dopamine neurons and how they're doing that. In a few years' time, we would love to be able to do experiments like that for MSA as well. So it's going to take time because we're at an information gathering stage for MSA, but it's a really important area, and it's being supported by the coalition. It needs more support.
Thanks. Larry, can you read the next question?
Yes. Next question comes from Huawei. Says, is there any good medicine for Parkinson's-like tremors? For this symptom, my dad, an MSAC patient, has already taken, he's got several medications listed here, and he says effects are not obvious. So any response to that particular question?
I mean, it's tough to give you a detailed answer on that one because, tremor can take different shapes and sizes. One of the types of tremor that can sometimes happen in MSA patients is a cerebellar-based tremor, and it's a little different from Parkinson's. It tends to happen more when you have an action with your hand, the tremor comes out a little bit more, whereas in Parkinson's, typically it's a tremor that happens when you're at rest. And so it can be a very difficult tremor to treat. Sometimes we use medicines that were invented for anti-seizure, anti-epileptic medicines that can sometimes quieten down those tremors. And so I think if you work with your movement disorder neurologist to think about, other types of medicines that aren't necessarily related to dopamine treatment, sometimes you can get some success with those things. So I would say look at those things and make sure the tremor is it a Parkinson's tremor, or is it an action tremor, or is it a cerebellar tremor? And in answering that question sometimes can guide the treatment choices.
And I see a couple more questions here about stem cells. Any hope for stem cells to reverse progression, not just slow or stop progression? And she also asks, is there any compassionate allowance for those in late stage to try stem cells? Dr. Singer?
Mm-hmm. So I can really answer this question only in terms of referring to mesenchymal stem cells, which is the only type of stem cells really that has been entered in clinical trials. And I can tell you from our experience with this intervention that reversal of already existing deficits is not something to expect from this treatment. We have had the occasional patient where there was one symptom somewhat improved 3 to 6 months after exposure to that therapy, but there's fluctuations of the disease, and all in all, the best we have seen is disease stabilization or slowing of progression. That does not mean that there's no hope. Stem cells, I think, are a big bucket of different types of cells that are yet to be explored, and I think that more advanced approaches where we actually induce these cells to become different types of cells that are actually needed in the brain. And if we find a way to help these cells make connections and get them to the right place in the future, that is certainly something that would be very much of interest, but we are not there yet. We're really not anywhere near there. I think Vic can answer in that regard, because he has done some work with those types of cells.
And I see another question. Is it useful to inject stem cells into blood vessels instead of bone marrow?
So to answer that question, the Korean trial that I mentioned before did utilize intra-arterial injections of mesenchymal stem cells, and they injected them in the carotid arteries in the neck and the dominant vertebral artery in the back of your neck to literally embolize the brain with these cells and randomly make those cells deposit into the various areas of the brain. They reported a positive trial. I cannot argue with that. At the same time, though, up to 30% of patients showed lesions on MRI imaging subsequently that looked like mini strokes. Most of those were asymptomatic, but it appears that these cells can actually block small blood vessels and therefore cause damage in the brain, which is why we pursue the intrathecal route, in other words, injecting those cells into the spinal fluid, where they also have more widespread access and can access not only the brain, but also the spinal cord, which we know is significantly affected in MSA. There's also a number of groups, oftentimes outside of trials, that are utilizing intravenous stem cells. That is a practice that I strongly advise against. The evidence for that is literally zero. And secondly, in theory, these cells would end up more likely in your lungs and in other parts of the body than the brain. Those are oftentimes the type of approaches we see commercially available, either just across the border or overseas, where treatments are offered for MSA, but oftentimes usually also many other neurologic diseases for a price. And I warn patients about those practices where really there's been treatments offered that are not actually treatments yet, that are still to be researched. And I think there's more risk to that than hope for benefit.
Larry, next topic.
Right. This has maybe a couple of levels of answers. Why does it take so long to get an appointment with a neurologist? My husband should have been diagnosed a year earlier. Now, there's a lot of answers to that, but could you maybe help a little?
You gonna make the guy from Tennessee talk about it? Well, I think in the United States, there's, I don't know where this question comes from, but at least in the United States, if you look at a map, you can see certain cities have a ton of neurologists, like too many, like Boston, and other people, there's no neurologists. So we call those neurology deserts. So in the Southeast where I live, there's neurology deserts in parts of Tennessee and South Carolina and parts of North Carolina. And, you know, it's a problem with healthcare, you know, across the United States. Hopefully we need to train, you know, we'll train more people. I think part of the MSA Coalition goal is to create these centers of excellence, which is where we have a recognized center that has dedicated their efforts and their energies to MSA care. And our hope is that those centers of excellence will become beacons of hope for local and regional populations around them. And I think we're looking to make these centers hubs for research, hubs for education, and hubs for clinical care. So, so stay tuned. But I think with the MSA Coalition, if you would like to partner with us in that, I think we'd love to have your support in helping us frame these centers of excellence and helping us to bring this opportunity to at least to North America.
Absolutely.
Yes.
Here's a question maybe Donna can take. When looking at genes, how certain are you that people in the database actually have MSA?
Thank you, Pam. That's an excellent question. And in fact, we are receiving data from patients with possible, probable, and definite MSA. So our database definitely has all of these diagnoses available. As Professor Wenning mentioned before, unfortunately, the definite diagnosis of MSA happens in autopsy-confirmed samples post-mortem. So we do have access to such samples, and those are the samples that are really most precious for the research and the discovery. So in order to find potential gene disease association, we would like to focus first on the autopsy confirmed samples that can provide us with some insights. And then obviously our goal would be to look at the less certain MSA diagnosis, like the probable and possible, and see whether we can find any links between the genes that were identified in the definite diagnosis and our population with less certain diagnosis. That could help us with diagnosis of patients and also with patient certification to see which patient could benefit from a different approach based on their genetic profile. That said, the challenge is that MSA is rare disease, as Dr. Khurana mentioned earlier. So it's hard to get to the number of samples with definite MSA diagnosis that provides enough statistical significance to make strong gene disease association. And that's why I think that the Core G, the genetic score of the MSA Coalition, is such a wonderful initiative because it truly helps a global outreach to places, neurologists and patients, where we can get the sample size which is required and needed to make these discoveries. I hope that addresses the question.
I'm going to just add one thing because I see a question a little bit lower down that's very related to what Dana was just talking about. I think a patient has come in with a question suggesting that they have a diagnosis of MSA and that it may run in the family. And is there any center that's looking clearly at MSA running in families potentially? This has become a very hot topic for the community now. As many of you know, most of the time we think of MSA as not running in the family, but we've seen a number of reports over the years, including 2 in just the last 12 months on multi-generation kindreds that have different types of synuclein diseases, sometimes MSA. And so we are very interested in those, and you can reach me through the coalition. But the infrastructure set up with Core G is now open to families. In fact, particularly well suited. The analysis platform that Dr. Vuzman has developed is very well suited to study pedigrees, potential familial cases. And so there's a mechanism now where we can sequence the genomes of patients who have this potentially in their family or a number of different neurologic diseases that seem suspicious for a genetic cause. So please reach out to us. We would be very happy to take your inquiry and triage it appropriately.
I have one here. Oh,
Yeah, go ahead, Larry.
I was going to say, I have one here. She's asking in several different pieces. Can patients further advance participate? Much can be learned even if a drug does not perform as intended. And I know it's been a question all of us have asked at one time or another. Can patients further advance participate in clinical trials?
Yeah, that's a good question. So I think behind that question is maybe a little bit of frustration that says, the inclusion and the exclusion criteria don't align with my clinical symptoms or my clinical severity. And that frustrates me because I want to be a part of this study, but I can't. And I think, that's a really, it's a very real frustration when you want to be involved in a clinical research study, but the way the study is designed, it doesn't kind of match up to fit the profile of your symptoms and your current stage of MSA. So I think, I go back to what I say. I do think that there are different trials for different people at different times. And, one of the things that I try and do as a neurologist who's involved in different clinical trials is I try and encourage companies to do different things like an open label study, perhaps in more severe cases, so that they could at least get access to a drug to see how the safety and tolerability works, or how certain things, pharmacokinetics work in the drug. And so I think we want to do that, but there are some times when there's economic restrictions or there's challenges in terms of getting drug to everybody that makes companies make difficult decisions about inclusion and exclusion. So I think the simple answer is it depends on the trial, it depends on the company, it depends on the medication or the therapy. But as clinicians, I think all of us would say that our goal is to get therapies for everybody, and we all work hard to do that. And sometimes it doesn't work out, but there's hopefully another trial that might be suitable, given the increased interest in MSA and how many companies are getting involved right now.
And it is true that pretty much all trials, either in the planning stages or ongoing, do focus on early disease. And there's nothing more frustrating for a physician sitting down with a patient and screening them and having to tell them, sorry, you don't qualify because your disease is too advanced. That is a very unfortunate situation, not only for the patient, but also for the physician. It's something that continues to bother me after going home from work. This is obviously something that is a problem. But as I mentioned in my talk, there is a rationale for doing that. That rationale is that it has been shown that at a certain disease stage, and this is typically sort of the earliest disease stage we can diagnose a patient with any kind of certainty, that's when the disease tends to progress the fastest. Not necessarily progressing as of the disease, but as we are capturing it on our clinical assessment tools. And that clinical assessment tool basically grades patients by what they can and cannot do, either in terms of their daily function or on the neurologic exam. And as patients progress and get more severe, that tool has what we call a ceiling effect. In other words, once you have reached a certain level of severity, it is hard to detect further progression. And therefore, these trials all focus on early disease thus far, because we are limited by the tools we have available to assess patients and quantify how quickly you progress or not. It's difficult to overcome that other than sort of looking at the assessment tools we have and see if we can come up with something that has a broader range. And we're actually in the process, in conjunction with the Movement Disorder Society currently, to revise the unified MSA rating scale. And we will hopefully come up with a scale that is more suitable to also capture progression at later disease stages. But this is ongoing work.
The way we've tried to address this issue for our patients is to essentially recruit patients who are interested in our clinic into a longitudinal study in which they're studied in detail from the perspective of clinical and biomarker imaging, and even in our center, generation of stem cell tools from our patient stem cell models. What we hope to achieve by that, and we've done this now once, is we execute clinical trials in parallel to the trials that are mainstream trials, the ones we've been discussing, that are really, for good reasons, focused on early patients, because we think we can learn a lot about how the drugs work and how they engage their targets. And some companies, but actually very few, are really interested in learning more about the mechanism of action of their drugs so that they can continue to design better trials. This is one way that we've found we have been able to recruit different types of patients into studies in order to learn more about the therapy. And I think that that's been very fruitful for us. We've actually helped a couple of companies understand their therapies a little bit better. And that's the model that we've been using. Again, as others have said before me on this, just answering this question, it's not for every company. There are many considerations, but sometimes it works out. And I think we can learn a lot from this method as well.
Thanks. Lucy asked, do any of our experts agree that a patient could have had MSA 20 years earlier than symptoms appeared?
I think there's a lot of evidence that there are prodromal stages to neurodegenerative diseases, Alzheimer's, Parkinson's, MSA, and you know, it depends. It actually becomes very difficult for us to really pinpoint when a disease process starts. I can tell you that if I make stem cell models from patients who have alpha-synuclein mutations, even a few weeks after I make neurons, I can identify at least vulnerabilities in their brain cells that I study in the lab. These are fetal stage cells that show me that these cells are abnormal and are set up for vulnerability and neurodegeneration. So that's really early stage. That's when patients have mutations. And I think we know now that for diseases like MSA, we talk about early autonomic features, disturbances of sleep, there are many symptoms that occur before those motor symptoms that suggest the disease process can really start a lot earlier than when a patient presents to the neurologist. And that's exactly why we're focused on early diagnosis and capturing patients as precisely as possible early on, so that we really have a chance of changing the outcome for those patients.
I guess my turn next.
Yeah.
Christine is asking, maybe it could be answered from a broader as well as a specific example, but my husband has been diagnosed with MSAC. During the initial symptoms, he was tested for Lyme disease and tested positive. Is there any link, or may having Lyme made him more susceptible to MSA? So maybe Lyme and then maybe autoimmune diseases. I don't know. Is that something we can answer?
Anyone want to take that? I can take it if you want to. There's no clear relationship between Lyme disease and MSA. Unfortunately, many people are treated even sometimes for protracted periods for Lyme when actually they have MSA. I've had a number of patients like this from the larger New England area that have been treated like this, and I think it's unfortunate because it delayed the diagnosis by a long period, and it gives patients false hope. So I think this connection is really not scientifically established and can sometimes be damaging to the patient's care. So that's kind of my blanket statement about that. Now, whether there are connections between autoimmune mechanisms, immune mechanisms, and the disease, I think is a very interesting one. There's certainly small-scale studies suggesting that dampening the immune system, for example, with intravenous immunoglobulins, IVIG, may be beneficial in some patients with MSA. And we know some of the patients have elevated immune markers, but really this is not definitively established. This is a very rare disease, and it's hard to make these connections. So there are some possibilities out there that need to be much better studied. The overall idea that infection and immunity play a role, the microbiome plays a role in brain health. These are very active areas of research and very important areas of research, but we don't have clear answers about their connections with MSA at this time.
Thanks. Next question from Sally. I saw my first symptoms in the spring of 2018, diagnosed fall of 2019. A month later, I was at my first conference. We took what they said and changed my diet to mostly Mediterranean, exercising daily, trying to keep stress to a minimum. Do you think this is why I'm still on my feet?
It might be. It sounds like you've got a great attitude on life. You're taking charge of your clinical situation and you're doing wonderful things. So keep it up. I think that we know that people that stay on their feet and exercise and take care of themselves do better. So thanks for being an example.
There's one more here from Grace, and this is one we all could answer, but there's some other places as well. Is there a central place to find out about clinical trials that are open to enroll?
I think the MSA Coalition website is a great place to start and get an overall perspective. Our patients can sometimes find it a little difficult to navigate the NIH website, but we tend to take our patients through that and show them how it works, which is clinicaltrials.gov. And as long as you put the appropriate search items there, that's a very useful resource. And maybe Wolf and Daniel know other resources too, but those are two places where I point my patients to first.
I agree, Vic. There's one comment I wanted to make on that, which is something that I did not realize until fairly recently, which is that one believes that the clinicaltrials.gov website is controlled and carefully reviewed in terms of the scientific underpinnings of the trials posted there, and that is not the case.
Mhm.
Pretty much anyone can post a trial there as long as they provide the necessary information that is postulated there. And there is trials on there that are called patient-funded research, and once the patient is enrolled, a bill comes of $10,000 to $20,000 for trial participation. That is obviously not ethically proper research, and one has to still be careful when browsing that site.
Completely agree. Thanks.
Yeah, thanks, Dr. Singer. So just want to put a plug in the MSA Coalition website. You could go to msacoalition.org. If you go to the bottom of your screen, there's a link there to the MSA treatment pipeline. So we try to keep that updated as much as we can to highlight the main trials that are currently underway. You also should subscribe to our newsletter. And again, at the bottom of our website, there's a link for you to subscribe to our newsletter. When we hear about new trials, we do send that out in our monthly newsletter. So just please keep in touch with us, and we'll pass along all those details whenever we get them.
I did have one question. It's not on here, but it has come up through the call line. There are, in several of the clinical trials, there's a cutoff age. And these people have heard about the idea of late onset MSA. They're calling and they're saying, I'm still able to walk, I'm still able to meet inclusion criteria, but I'm too old, based on what the criteria says. Is there going to be any thought to extending that age requirement? Is there a reason why 75, 78 are ages where the cutoff age is placed in that clinical trial? That's what they'd like to know.
I can answer you my thoughts on it.
I think oh, sorry. Go ahead, Will.
Sorry, Dan.
We'd like to hear your thoughts. I was just going to say every trial is different. There's not like a one cutoff. That's what I was going to say.
Yeah, it's different from trial to trial, but it is an age cutoff, and I think there's two reasons for that. Number one, we do know that at higher age, the rate of misdiagnosis is likely higher. That's, I think, one concept. The other thing is actually some guidance we received, and we submitted our own trial for NIH review. And the reviewer was concerned that if we do not restrict the age, that we will see a lot of co-pathology, prodromal Alzheimer's disease or other neurodegenerative diseases, adding to the brunt of deficits in making the assessment less clear or less clean. I think those are reasonable thoughts as to why there's a tendency for age restrictions, but I'm curious what others are thinking.
I see a question that just came in. Are there any other tests available now besides the SIN1 test that some places offer? Maybe someone can talk about that SIN1 skin test and what that actually tells you, and if there are any other tests similar to that people can get access to.
Mm-hmm. Right now at our center, we use the SIN1 test quite a lot. We've had So I'll tell you a little bit about the test. There is strong evidence that the types of proteins that aggregate in the brain also aggregate elsewhere in nerve cells in the body. And so one place to look at nerve cells that's quite easy is in the nerves that are really beneath the skin. And so with a small skin biopsy, you can take a look and microscopically look for the protein that you think might be involved in the neurodegenerative process in that patient. And for synucleinopathies like MSA, that protein is alpha-synuclein. And so this test is quite sensitive and specific as far as we know. And it's become quite helpful because it allows us to quite definitively say whether a patient has an alpha-synuclein disease or not. I think we need more data on the test, and we will get more data in the next couple of years, but the early data looks very good, and we have certainly found it to be a good tool for diagnosis in our clinic. Wherever we have had our patients generously donate their brains after death, and we've had those tests before, they have always been concurrent and concordant, I should say, in consistent with the literature. So that's been a very useful test, because sometimes you can get a patient who doesn't have Parkinson's disease, has some kind of Parkinsonism, and you want to distinguish a synuclein disease from another disease like a tau disease, like progressive supranuclear palsy, and this test is useful for making that distinction. Right now, it's favorable in terms of insurance and coverage, and so we have had a lot of luck in patients not needing to pay much out of pocket for this test, but you'll have to talk to your own physician about that and see how appropriate that is for you at your center. There is another test that looks at spinal fluid and looks at alpha-synuclein in the spinal fluid. That's a slightly more involved test, but it has some advantages in that you can really look at the different ways in which alpha-synuclein is aggregating, and Dr. Singer has played a great role in some of that research. And there's a company, Amprion, that is working on, has really commercialized that test. But that's still, as far as I know, maybe Dr. Singer can update us, but there's still a fairly high out-of-pocket cost for that test, at least for our patients, that's been the case. And so we have been using the SYN-1 more, but we eagerly await these different types of tests. And you can get synuclein from nasal brushings and other places too. So we're probably going to see a great expansion in this set of tests to diagnose synuclein diseases. But maybe Dr. Singer?
We have a little bit of different approach to utilizing these tests. You do not use those in clinic. And there's a simple reason for that, which is both tests still have to go through further validation stages. And particularly the SYN-1 test currently is based on research that comes from one area, from one center. And all the other research on alpha-synuclein in skin results look different. The current validation trial is still going on. And I think personally, also seeing patients come to clinic with not a hint of alpha-synucleinopathy who had a positive test done and done for some reason is very problematic, and we've struggled with that in our own clinic.
That's interesting.
And so with all that, I think we should be careful. These tests are very promising. We're very excited about them, but let's go through the proper validation first, and then utilize them. That's at least our approach at the moment.
I think I should address that. I think that's a great point, and that's why I also believe that more research needs to be done on those tests. I think we never use one test in isolation. I think that's really important for me to emphasize, and maybe I should have. We use a whole battery of tests, very much in line with the new consensus criteria. And if in that context there's a consistent result, we take that as highly informative, and it has been informative, at least in any case that's come to postmortem. But I think you make a good point.
I'll make one extra point in that SYN-1 test is not a diagnosis of MSA. It's not a test that's going to tell you if you have MSA or not. And the other point I'd make is, in our hands, about 5 to 10% of patients might have a positive SYN-1 test, but have a negative CSF amplification assay test. And so we don't know what to do with that. So I think the one theme that you've heard from the other two neurologists is that the clinical exam matters. And so a really good clinical exam is almost the better test sometimes than a lab. And I think going to see a movement disorder or on an autonomic team can really help get the diagnosis correct.
Again, the test is a test for synucleinopathy, not for MSA, so it has to be put in the right clinical context. And we know that many patients come to post-mortem and we look at their brains. This is in population-based studies of aging. And we'll see, for example, a disease called incidental Lewy body disease, which is that there's alpha-synuclein in the brain. but those patients in life never had a clinical disease. And in the same way, alpha-synuclein can deposit in different, as I said, different regions and nerve cells. And so incidental disease in the nerve cells that you see in a skin biopsy doesn't mean you have the brain disease. And so I think this is a complex area, and you do need to use it in conjunction with other tests and the clinical examination and history.
Okay, thank you. On that note, we're unfortunately out of time, and I wish we had more time to answer your questions. Please, everyone, if you have questions we didn't get to, you can send them to info@multiplesystemattrophy.org, and we'll try to get you answers. I want to thank everyone for your participation, everyone who sent us questions, and for our wonderful panel who took their time on a Saturday to be with us. We're so grateful that we have your expertise. And thank you so much. Thanks, Larry, for helping moderate.
Thank you all.
Everyone, and I hope you enjoyed the conference. Please look at the replays, and we hope to see you in person again, maybe next year. Let's hope. Bye now.
Thank you.
All right.